to present for us. She has a master's in epidemiology. She's a former Fogarty scholar, and she's NIAID staff overseas stationed in Cambodia. So when Cambodia identified its first patient with COVID-19 in late January, Jessica Manning quickly sequenced the genome of a virus sample and deposited it on the next strain, which is the Global Collaboration Database. It was among the first 20 to be shared on the site, which now includes thousands of SARS-CoV-2 genomes. She says it was a really big step for them to be the first lab from a developing country to contribute to the global knowledge database. And she'll talk about her projects in COVID-19 briefly after the presentation. Given the growing global incidence of mosquito-borne diseases such as malaria and dengue, Jessica's studies aim to use genomic sequencing to identify pathogens in samples collected from people suffering from fevers. She has funds from the Bill and Melinda Gates and the Chan Zuckerberg Initiative. Her main research focus is investigating the potential for a universal vaccine for diseases transmitted by mosquitoes created from the insect's spit or saliva. We know that, she says that we know that saliva actually worsens disease, so if we can have the body mount a protective response against saliva, we may be able to attenuate the actual pathogen's effects, be it dengue, Zika, malaria, whatever is carried by the mosquito. So thank you very much, Jessica, for being with us here today. She's speaking from the East Coast in the USA, so she's 1 hour before us, very early. So thank you. I hope you have I hope you enjoyed the presentation. Welcome.
Okay, good morning. Thank you, everyone. I'm just going to put my presentation up briefly. All right. Okay, great. So thank you, Camilla, for the introduction. As she said, I am an infectious disease physician scientist based in Cambodia, normally, with the National Institute of Allergy and Infectious Diseases. And our field sites and laboratories in Cambodia are mainly focused on mosquito-borne diseases, but more recently, like everyone, we've had a bit of a COVID-19 shift. However, today's talk is going to be on our first in human mosquito saliva vaccine. And this is work that was done at the NIH Clinical Center in Bethesda before I moved to Cambodia permanently. And what I would like to do first is go through the rationale of why we would think about using salivary components in a vaccine, talk briefly about the work that was done at the bench on this particular vaccine, and then how we brought that forward to the bedside. And really the whole reason this vaccine made it from the bench to the bedside in such rapid fashion, the first time ever we've been able to use vector salivary components in humans, was because of the Zika epidemic, obviously that Brazil bore the brunt of. And then how we're now translating that from the first in human trial now back out to the field work in Cambodia. So to start, let's see. All right. So the rationale, these are mosquito salivary glands right here, of the why and the how we would be using mosquito salivary glands. Let me just make sure this is going. Yes. All right. So I don't need to probably tell this group in Brazil that we know that the likelihood of vector-borne disease in humans is going up, and that now it's making up words of 17% of all communicable disease, although maybe SARS-CoV-2 has shifted some of these statistics in the last several months, but causing at least 1 billion cases per year and 1 million deaths per year. So let's see. There we go. So the question is, particularly, again, in the setting of emerging viruses, but also emerging viruses and emerging pathogens and new vectors, is this our new reality? And how can we be better prepared? And as the year 2020 has shown us, we're not always very well prepared when a new virus emerges. But if a new virus or a new pathogen emerges in a vector, what could we possibly do to be better prepared without having the knowledge of which pathogen it is? And so excuse me, I'm having some issues. There we go. And so one thought is that we can target the common element for vector-borne diseases by targeting the vector, right, which is the critical piece of the transmission puzzle. And so, excuse me. And so that would be that vector saliva, in fact, drives infection. And so if we're capitalizing on the fact that if we can take saliva, and I'll explain again why, could we in fact have broader vector-specific protection? So as Camilla mentioned, you know, a universal mosquito-borne disease vaccine would be amazing, but really at what level could we think about achieving protection? At the family level, the genus level, the species level? And even if we were able to basically capitalize on this just for one genus species such as Aedes aegypti, you can imagine the impact we would have given that Aedes aegypti is competent for so many clinically relevant pathogens and even more possibly. So we can think about this in terms of applications for emerging pathogen outbreaks. We can think about it in terms of travel medicine if it's not possible to actually vaccinate total endemic areas, or if vaccines don't offer us long-lasting protection, that would be more appropriate for travel medicine. Or also considering that this saliva components would be an adjuvant for a pathogen-targeted vaccine, such that you could combine a pathogen target and a vector target in the same vaccine, which they are thinking about doing right now in Europe with leishmaniasis and a sandfly salivary protein. So with that, as this audience may know, that saliva contains many active compounds and is a virtual pharmacopoeia of vasodilators, anticoagulants, immunomodulators with lots of different functions. And so what we want to do is begin looking at all of these different immune cells that are impacted here in the dermal epidermal environment and think about how we can capitalize on that in order to inactivate or make it less successful for a pathogen to establish infection in a human host. And so we have a large body of animal model evidence that shows that vector saliva increases disease burden, and that if you take it a step further and immunize with vector saliva, it actually protects against disease. And we don't see this limited to one particular vector pathogen pair, but we see it across many different types of pathogens and vectors: viruses, bacteria, parasites, as well as mosquitoes and ticks and sandflies. And I'm just going to very briefly go through some of the mosquito data. And you can see here that mosquito saliva can actually modulate the cytokines, which has been one of the mainstays for over a decade of how we think that saliva may be impacting the establishment of disease, because we see that Th2 predominant cytokines are actually increased in the skin when bitten by a chikungunya infected mosquito compared to a needle, as you can see here for IL-4 with the mosquito bite, and IL-10 compared to a needle alone. And then if we actually look at without the saliva, that we see interferon gamma expression increasing. However, we also know that outside of just looking at cytokines, that saliva can increase the dissemination of a pathogen and then the mortality in the animal host. So here we have a few different examples of different mosquito-borne viruses. So Simliki forest virus, which is an alpha virus, West Nile, and it was just a flabby, and then a bunya virus. And we can see here with Simliki forest virus that if we're in the dark bars, if the virus is distributed via the bite of a mosquito, that we have much higher dissemination levels in the brain. And that also if it's disseminated through the bite of a mosquito, we end up having much higher titers of the virus in a mouse. And then if we move on to bunya viruses, we can see just wider dissemination of the virus when the bunya virus is present in the bite of a mosquito, so with mosquito saliva present in the skin, spleen, and serum. And so, excuse me, everybody popping up and joining the meeting is throwing off the, the presentation a little bit. And then again, if we take this one step further and we think about immunizing with mosquito saliva proteins, that we can see in here with the example of parasite and Anopheles bites, we can see if we pre-sensitize or the animals are exposed to Anopheles bites beforehand and then they are challenged with the bite of an infected malaria-infected mosquito, we end up seeing much higher percent parasitemia than if they had not in the naive population. And so taking that a step further, again, we see in vaccinated mice with Culex saliva being challenged with West Nile, that we see that saliva-vaccinated mice have a much higher survival of 100% compared to mock-vaccinated mice at 30% survival. And so that we believe there's some level of protection that gets conferred if you've been immunized with saliva or you've been pre-exposed to mosquito saliva in some way. So, what would be the mechanism for this? So if we begin to think about what the hypothesis is for a saliva-based vaccine, we go back to the animal literature. And so using the example of viruses and mosquito saliva here, we can see that, the mosquito comes along, it injects the virus and saliva into the dermal epidermal center. We see the increase of Th2 predominant cytokines, a dampening of the interferon response, and that in and of itself brings in both the saliva and the pathogens, can cause a rapid influx of neutrophils, an increase of certain chemokines that are then going to bring in a wave of myeloid cells that are now submissive for further infection by the virus that track the infection down to the draining lymph nodes, and now the infection has spread. And so the thought is, if we can immunize with a super therapeutic dose of immunodominant salivary proteins, they get taken up by an antigen presenting cell. And here we're not exactly sure, but if that can prime a CD4, CD8 response in the lymph nodes, then perhaps the next time in the far panel, when a infected mosquito comes along, the human host is able to mount a rapid tissue resident memory cell response, being primed by that CD4 or CD8 response that causes increased interferon gamma, activated macrophage, basically creating an inhospitable environment for the pathogen that can block replication and dissemination, such that no active pathogen reaches the draining lymph nodes and they're able to control it. So where we were was, at this time, there was a whole host of animal literature that has been accumulated over the last 20 years in rodents and beagles and hamsters. And then in 2015, a colleague of mine, Fabiana Oliveira, whom you may know, and Jesus Valenzuela and others, did the first sandfly salivary antigen in rhesus macaques. And then in 2016, we saw the first biologically relevant kind of tick feeding saliva vaccine go into cows in Africa in the wild. And although that was not successful because ticks are a whole other beast, we thought that it was time now to really move forward from the bench to the bedside with a first-in-human mosquito saliva peptide vaccine. And this was initially all started in 2016 in response to the Zika epidemic, about having more tools available to us in the setting of emerging vector-borne disease pathogens. And then our trial was actually launched in 2017. So I'll go briefly the bench work. So the name of the vaccine was AGSV, which stands for Anopheles gambiae saliva vaccine. And from those of you who may have seen the Lancet paper already, what basically happened was, very typical steps of taking Anopheles gambiae salivary glands, running them out on SDS page gels, and then applying this T-cell epitope algorithm that was developed by a company called Seek to those particular different bands of molecular weight of the Anopheles gambiae saliva gland homogenate. And what the algorithm did was categorize the T-cell epitopes basically based on whatever proteins were in that band for its structural affinity for a peptide given its MHC, HLA allele, and reactivity. And what the algorithm is trained to do is to be looking for peptides that would possibly induce a strong Th1 response or an antibody response. And so based on the algorithm being applied to the different molecular weights of Anopheles gambiae, it identified 6 different peptides, and ultimately 4 that were put into a combination at 50 nanomolars each. And then in CD1 mice, these were then injected to see if they could induce an antibody response. And indeed, you can see here, we have a total IgG response in the vaccinated mice compared to the placebo mice. And then we look at the isotype of IgG2A. in the vaccinated mice. But remember, for a vaccine, we don't necessarily just want an antibody response, particularly if we're thinking about this Th2 to Th1 shift that we went through with our hypothesis. We're also looking for a strong Th1 response. And so in order to test that, the peptide mix was incubated with the splenocytes from mice, and then looking at what the interferon gamma induction response was in mice that were vaccinated, which is in the dark purple. And you'll see here Con A is our positive control, which worked in both the placebo group and then the vaccine group. And then looking at a couple different concentrations of the vaccine, we see that we indeed have higher interferon gamma release in the vaccinated group, which has now been stimulated with the AGSV peptides in order to basically get that interferon response release. So that was actually most of what we had in terms of preclinical data, except for some challenge data that showed some protection, not a slam dunk, but when you then gave vaccinated mice who had been vaccinated with the saliva vaccine versus, which is this AGSV mix here, versus if they had just been vaccinated with that particular band of the salivary gland homogenate, versus a non-related peptide mix, which you can see here. The placebo animals had a mortality rate of 50% after being challenged with a plasmodium-infected mosquito, compared to 20% mortality rates given the other modalities of using saliva vaccination. And so all of this together in the setting of trying to move this forward into humans, in the setting of the Zika epidemic, we then basically just went on to get some safety data in white New Zealand rabbits and then move this into our bedside trials. And so we called our trial SPIT, for Saliva Peptide Immunization Testing Trial. And the purpose of this was to be able to give that same subcutaneous vaccination that I just showed you for the preclinical data in the mice into humans. And typically for phase 1, these are smaller trials for those of you who aren't as familiar with clinical studies. And so we typically do 40 to 50 people in a few different groups, and we're only looking at safety. But in this case, we were looking at both the safety and the immunogenicity in each of the 3 groups. So for this trial, we had 15 people in the AGSV group, 15 people in the adjuvanted group. And so the adjuvant in this case was Montanide ISA 51, which is an oil and water emulsion, which will also drive a Th1 response. And then we have placebo, which was sterile water. And so we basically screened our healthy volunteers, we consented them, and then they were vaccinated on day 0 and day 21, again, subcutaneously in the arm. And then on day 42, we challenged them with a mosquito feed. And this was done for safety reasons, because we didn't want people to then go back to living their lives and get a mosquito bite in the summer and have this huge reaction. So, so we did a mosquito feed on day 42. But also we were interested in whether or not there would be any cross reaction between the Anopheles gambiae salivary peptides and Aedes aegypti, which at the time was the mosquito of interest given the Zika epidemic. And so this mosquito challenge was actually done with Aedes aegypti mosquitoes. And then we followed them up for 12 months because this was a first in human vaccine trial, and so the FDA requires this. So moving on to the objectives. So as I mentioned, it was a phase 1, so we were primarily interested in, is this safe? Can we give saliva components to people in these huge doses as a potential for vaccine? But also, is it gonna work at all? So immunogenicity in terms of what is the humoral response? Can people make saliva vaccine specific antibodies? And then number 2, what's gonna happen to their cellular immunogenicity? So we were specifically interested in what cytokines we could measure and whether or not those would be more of a Th2 predominance, Th1 predominance, when we took our vaccinated volunteers, PBMCs, and incubated them with the vaccine peptides. However, we were also interested in some secondary objectives after the mosquito feeding on day 42. So number 1, could we see any type of boost to vaccine-specific total IgG? And then again, what is their cytokine profile after being stimulated with these peptides? But we were also interested in what happened to the mosquitoes. So there had been some preliminary evidence in the preclinical data that perhaps the mosquitoes were not able to lay eggs as well or survive as long. But it was the data was not clear, and it could have been insectary mismanagement. So we, after the mosquitoes fed on the volunteers, we then followed them for up to 14 days to see, did they lay eggs after the blood meal? And then did they survive? And then did their progeny survive? So did those eggs develop into pupae and then live onwards? And so that was a secondary objective in seeing, well, could there be some type of transmission blocking component by following mosquitoes that had fed on vaccinated volunteers? So to the data. Oh, actually, first, who was in our trial? So we had 73 people screened, ultimately enrolled 49 healthy adults of predominantly Caucasian, African American, and a couple Asian American. And they were randomized to the adjuvanted arm, to the vaccine arm alone, or to placebo, which was again sterile water. And then each group had 2 doses. And again, we did this all in an intent to treat analysis. And then we had everyone go through the mosquito feeds, as you can see here. And then we followed everyone up for 1 year. But again, the primary objectives were all measured by day 42. So we didn't replace volunteers that dropped out after day 42. So to answer our first question, is a vector saliva-based vaccine safe? So you can see here in the top, we have our adverse events. And these are systemic adverse events, slightly higher in the adjuvanted arm, but nothing significant. What you'll notice is significant is here in the local injection site reactions. We did see significantly more adverse events right there at the injection site, mainly in terms of erythema, pain, swelling, which is not uncommon with the adjuvant we chose. But interestingly, you can see here that itching was significantly increased in the adjuvanted arm, as well as the vaccine arm here, which we could expect given that we were giving super therapeutic doses of the mosquito saliva antigen. So overall, we felt that this was a pretty good safety profile. We did have one grade 3 adverse event that you can see here, where after injection, this participant ended up with an 8-centimeter injection site pain, redness, and swelling, and then some systemic hives that we had to treat with steroids and antihistamines. So this was unfortunately the one volunteer who we then did not give a second dose of the adjuvanted vaccine to. However, she did undergo the mosquito feed and had no adverse events whatsoever and has done well since then. And again, as I just mentioned, when you remove the adjuvant, which we know can cause that redness and swelling, the vaccine alone was only noted just to have more pruritus or itching at the injection site than placebo. So next, if we give a saliva-based vaccine, can we actually produce vaccine-specific antibodies? And if we can, how long do they last? Would this really be a viable idea for a vaccine? So you can see here, the adjuvanted vaccine was able to produce vaccine-specific antibodies. So if we look at our primary endpoint at day 42, so again, you've received a vaccination on day 0, on day 21, and then we've measured your antibodies on day 42 prior to your mosquito feed. And we can see that mainly predominantly in the adjuvanted arm that we have increased levels of vaccine-specific antibodies. And we did this by looking at what was the change in optical density on ELISA, a ratio at this day 42 compared to day 0. And we took the geometric means. And then we can also see that this effect lasts out to day 102, again, only in the adjuvanted arm, excuse me. And that these were primarily IgG3 antibodies. And then we see that the effect goes away by about 1 year. So this was the last day that we had data from most of the group, day 332, and we see that the effect is completely gone. So moving on to the next question of whether or not cellular immunogenicity, can we see a change in cytokines when we stimulate our vaccinated volunteers, PBMCs, in order to produce interferon gamma, predominantly Th1 cytokine versus others. And so what we end up seeing is really a mixed response here. So again, we take peripheral blood mononuclear cells from our volunteers who either received the placebo, the vaccine, or the adjuvanted vaccine. And we stimulate them either with the vaccine peptides as compared to a negative control. And so you see this ratio here of when they're stimulated with the vaccine versus our negative control of media. And then we also, for all of these, although I'm not showing it, they had a positive control to Con-A to make sure that they were working on our Luminex platform. And so we were encouraged to see, as we had hypothesized, that the vaccinated volunteers would have a higher response to have a higher release of interferon gamma. So as you can see here, at day 42, we only see this in the adjuvanted group. But by day 102, whether or not there's a maturation of the immune response, we can see that there's more interferon gamma released in both the unadjuvanted and the adjuvanted vaccine groups. And that was really the main thing that we wanted to see, that we could get some type of shift in our Th1 predominant cytokine interferon gamma. And then other than that, we ended up having kind of a mixed response, given that we do see an increase in IL-5, which we see more as a Th2 response, and then GM-CSF. And then for IL-10, as well as all of these cytokines over here, IL-2, TNF, IL-4, IL-1 beta, we did not see really any response at all, or any different changes between our three different groups. So we were mainly focused on, for this, maybe the only strongest difference that we can pull out is the interferon gamma release. And that perhaps these other cytokines that we would expect to see are really difficult to study in PBMCs in vitro and would be maybe better studied in the dermal, epidermal environment. So moving on to our other objectives, as you remember, we wanted to see what happened to the mosquitoes after the day 42 mosquito feeding. And looking at this data, I can say we did not see that there was any change in mosquito fecundity or survival between the groups. So I don't have the data here, but basically we watched all the mosquitoes took a 100% complete blood meal on all of the volunteers, regardless of if they've received vaccine, adjuvanted vaccine, or placebo. All of the mosquitoes laid eggs without any problem. And then we were looking to see if there was any difference when that progeny grew into pupae, and then those pupae grew into adults. And again, we don't see any significant difference between the three groups, as you can see in these two different box and whisker plots. So it did not appear to have any type of onward effect on the mosquito or the progeny. So our take-home messages from this were really about moving saliva-based vaccines into the clinical space, and that they could be safely administered in humans. We had mild injection site reactions, mainly when adjuvanted, and that, indeed when adjuvanted, we could see that there was antibody production to our vaccine, our saliva peptides, and that we could also see an increase in the interferon response, but otherwise there was a mixed cellular response that wasn't clearly Th1 or Th2 predominant. And so again, are we only seeing this mixed response, cellular immunogenicity response in the PBMCs, or is this a local phenomenon in the skin? And so we're currently planning follow-up studies to answer these questions, and to also just better understand and refine that hypothesis of how saliva is actually working in the dermal epidermal environment in humans, and trying to extrapolate and continue to move the lessons we've learned in the animal literature onwards. And I will say, lastly, at the bottom here, there is, as a follow-on, what we call AGSV+, which was contracted out to University of Maryland, which a fifth peptide has been added to the AGSV mix instead of just the 4 single peptides. And skin biopsies were also being done as part of this phase 1 vaccine, but sadly, because of COVID-19, this has been kind of stalled at the moment. So we'll have to wait and see what's going to happen with the HASV plus. But in the meantime, kind of moving to these big questions of how saliva mediates pathogenesis, we basically go to the field. And so this is Cambodia in Southeast Asia, where I'm normally based, not on the East Coast. And here, like Brazil, Cambodia is hyperendemic for dengue. And, our thoughts were, if we've been able to show this proof of principle with Anopheles gambiae, why don't we use a different approach to think about the same process in Aedes aegypti mosquitoes? Because obviously Aedes aegypti carries some of the most clinically relevant arboviruses: Zika, dengue, chikungunya, virus. It's competent for Rift Valley fever virus. So if we can think about doing this with Aedes, certainly that would be a good place to think about moving saliva-based vaccines. So we established a cohort vaccine called PAGODAS, a pediatric assessment group of dengue and Aedes saliva. And in that, we're actually trying to use these vector-borne determinants of disease in Cambodia by first just setting up, plain seroprevalence studies to dengue, as well as understanding what is the Aedes aegypti salivary antibody intensity in these children by sampling them every 6 months, wet season, dry season, wet season, dry season. And then we also follow them for dengue rapid testing, other Aedes aegypti testing, such as chikungunya and Zika. And then, as Camilla mentioned earlier, some of our work with Gates and Chan Zuckerberg, we perform metagenomic sequencing on all of our febrile patients, both in the cohort and in our different cohorts for emerging viruses like COVID-19. And so we initially basically set up that we did indeed have enough of a burden of dengue in Cambodia, and that we predominantly see it as children age. So we were interested in these more naive populations becoming immune to both dengue and Aedes aegypti saliva, so that we could understand what those differences are. And so we do know that dengue 2 is the most prevalent serotype in Cambodia. And now that we had established kind of baseline disease burden, we began looking more closely at how the saliva intensity aligned with basically being able to predict where our outbreaks would be. So in 2019, Cambodia had the largest dengue outbreak they've ever had in history. And we were basically using 2018 saliva levels able to predict where those 2019 cases would be. And you can see that by looking at where there's more intense transmission and then counting up where our symptomatic cases were. And this is in our field site in Cambodia. And so with the development of disease, we're able to begin to characterize that systemic immunity and begin to think about biomarkers and vaccine candidates for Aedes by looking at immunodominant Aedes aegypti specific salivary proteins, begin to define those kinetics, because remember, we saw the Anopheles gambiae peptide vaccine basically trail off by 1 year. And so what's happening in the field in people who are being bitten year round, and then how can we begin to characterize some of these cellular responses to saliva in our cohort when they're at these different stages of their dengue infection. And so I'll briefly mention, basically moving forward some of our immunodominant proteins where we're looking at high, middle, and low responders to Aedes aegypti saliva, and then looking at the different bands, which is the same approach taken with the Anopheles gambiae peptide vaccine, by then characterizing the peptides that we see most frequently in our cohort responders, making those recombinant proteins. And you'll see that James and Fabiano here are mainly responsible for this work. But we've identified 5 different Aedes aegypti salivary proteins to move forward and characterize, because we can see that they respond very well in our Cambodian cohort. When we compare them to our clinical center volunteers in Bethesda at the NIH, we don't see as much reactivity, which we would expect, obviously, given that these folks are living in Bethesda and not living in Cambodia. And then right now we're in the process of basically using paired samples and beginning to understand the patterns of salivary protein antibody reactivity in the setting of disease or no disease, severe disease, asymptomatic disease. And this is really kind of where we're trying to move forward, as well as begin to characterize some of these epitopes for the possibility that we could move some of them forward as NIAID's Egypti salivary-based vaccine. So with that, I would like to thank our big team in Cambodia that you can see here. And then also our teams in the clinic and in the field. These are the pagodas where we work in our community-based studies. And then also all the other people who have made the vaccine possible at the NIH Clinical Center, as well as all of our work in Cambodia possible. So all of these folks here at the NIH, as well as SEEK for the algorithm and production of the vaccine. And then Chan Zuckerberg and Bill Melinda Gates for some of our COVID-19 work. And Camilla asked me just to say a few things about our work on COVID-19. So you can see here, here's our laboratory, here's our pagoda, and here's the clinical center. So our COVID-19 work in Cambodia was mainly, we were, I think, the 5th or 6th country to have a case of COVID-19 in January. And we knew that was coming because Cambodia receives direct flights from Wuhan nearly every day. And so we had time to basically be a couple weeks to prepare to know that we would want to be able to sequence the virus and contribute to the global genomic epidemiology efforts in order to track the virus. And fortunately, we have cross-sectional cohorts of febrile disease set up specifically for metagenomics. And so when the first case came, we were able to basically extract the RNA and then put it through our unbiased metagenomics pipeline. And when we did this, our pipeline is called ID-Seq and it's hosted by the Chan Zuckerberg BioHub. We actually saw that it had a 99% homology hit for a SARS-CoV related virus, because we actually, at this point in time in January, there hadn't been an NCBI update yet to SARS-CoV-2 after the Chinese researchers had deposited their first sequences. And so we basically had proof of principle that this unbiased metagenomics platform could identify emerging viruses because it did identify that we had on our hands a SARS-CoV related virus that we then went on to confirm as SARS-CoV-2 and deposit into the into NextStrain and GIS-aid databases, which now have thousands of viruses. And there's been some really amazing genomic epidemiology work done by a lot of groups now. So now our COVID-19 work has mainly been focused on doing the sequencing whenever we've had outbreaks in Cambodia in February and March, so that we can do some of the transmission chains. So when we have a particular cluster at a Chinese casino in Cambodia, or with a Muslim pilgrimage where there was an outbreak. And now that the virus is under pretty good control in Cambodia, we're moving towards doing seroprevalence studies for COVID-19. So we will be launching in the next month or two, basically annual surveys of 2,000 people at a time in Phnom Penh using just a basic ELISA to the RNP and the spike proteins in order to see whether or not Cambodians were exposed and just didn't develop symptoms, or whether or not we really have done a good job of keeping COVID-19 at bay in Cambodia. Because again, it is a very open air society. The median age of the population is 25. And so we haven't seen as much of the SARS-CoV-2 morbidity mortality that has been seen in the US and in Europe and Brazil. So with that, I think I will pause here and be happy to take any questions about our saliva vaccine or about SARS-CoV-2. So thanks everyone for listening.
Thank you, Jessica. It's really fantastic presentation, really, really interesting. Just a quick comment before I start with the questions. It seems like you're living in our pre-world, because you're saying there's no COVID case. You were talking to me before. There are no COVID cases in the last 15 days or so. So it feels like you're talking from an ancient world, you know, where you can go about your life like you used to. So that was really interesting. It was really fantastic because we could see our work from the beginning, from the lab work, the first evidences, and then moving on to the bench work and then the field. It's really amazing to see all this evolution. So I'll start with the questions with one of my own. It was interesting that when we look at the antibody response and the cellular response that you see in your volunteers, that with the antibody response, you see a really important effect of the adjuvant, whereas for the interferon gamma response, the adjuvant doesn't seem to really play an important role because you have same levels. So that was, can you comment on that? That was interesting, I thought.
Yeah, so some people, I mean, you know, obviously now specifically, as we've all been racing for vaccines with SARS-CoV-2, we've been thinking about all these different platforms, right? mRNA vaccines, DNA vaccines, peptide vaccines, and then, you know, the old way, right, where we're using attenuated pathogens through some type of recombinant technology. So I think that a lot of people think that with a peptide vaccine, you're not going to really get any type of immune response if you only use the peptide. And so you really had to have the adjuvant in order to get that immune response, to see the humoral immunogenicity, to get the vaccine-specific antibody response. And so some folks are really just not surprised at all that we see not that much antibody response with the unadjuvanted peptide vaccine. So I think moving forward, one way we could think about improving upon this, so let's say we find proteins that are of interest to us with Aedes aegypti, think about taking the entire recombinant protein and of interest instead of just a single peptide that was identified in silico, and putting that on some type of viral vector so that you would get the, like, let's say a modified Ankara virus, right? So then you would have the Th1 response being driven by the viral vector, but it has a payload, and those viral vectors can carry payloads of up to, 80 kilodaltons. And then you can put that on, and then that may be your way around the fact that maybe the peptides in and of themselves can't stimulate enough of a humoral response alone. But we did see enough of the peptides to be able to show that without adjuvant, we could still get interferon response. So they clearly retained some type of Th1 response.
But if you're thinking that if you think that the antibodies are to the salivary proteins, and what you're looking for from your diagram there, you're looking for the cellular response, maybe not having the adjuvant, and specifically when you show the side effects, so to speak, maybe it's enough just to have the cellular response, because these antibodies are to salivary proteins and not to the pathogen, right?
Right. And the question is also that, if we were to use this in an endemic area, could you get natural boosting of a saliva-based vaccine just by natural exposure to mosquitoes? Or do you really need that super therapeutic dose of antigens? And I think that's the question. And I see another question from Claudia, which I actually just mentioned, but this was a really good point about nanoparticles, virus-like particles, or adenovirus. So that was, what I wanted to mention was, I think moving forward, that we have, number one, we have better adjuvants to choose, and we have better vehicles. And I think, adenovirus, like I said, modified Ankara virus, thinking about using those viral vectors and then putting a salivary protein or peptide on that is going to be, I think, a better way to go for the future. And then also for adjuvants, Montanide is pretty cheap because it's old. Okay. And some of these adjuvants that are used in vaccines can be expensive because GlaxoSmithKline and other private companies license them. So I think if there was going to be a real move in the future, that it's going to be using, some type of alum type adjuvant, because we know that those are typically better tolerated than Montanide.
And how much does the baseline response to the vectors that you have in Southeast Asia, for instance, how much would that impact once you have a vaccination schedule? How much of the pre-existing immunity to these salivary proteins?
Yeah, so that's a really good question. So we've started with the children, and we're moving into the adults, right? Because the question is going to be whether or not these children have really robust antibody responses. And we certainly see them more in the younger kids. And then we see the tolerance, right? And so then we see the tolerance in the adults. And so I didn't mention it, but we've also recently started a skin biopsy study where we're feeding the mosquitoes on the Cambodians and our Cambodian volunteers who are so exposed to these vectors year round, and basically seeing, is it anergic, right, in the skin, or are we actually still seeing responses? Because if you are now 50 and you've been exposed to these salivary antigens your entire life, and now you're just tolerant, you're completely anergic, then that would just defeat the purpose of ever considering a salivary based vaccine in an endemic area. You would only be reserving it, right, for travel medicine, people who were naive to a particular area. So that work is underway, and we're just going to have to see how your baseline either tolerance or reactivity impacts the way you you respond. And we're using a few different modalities for that. So after we take the skin biopsies, we do flow, RNA-seq, TCR-seq, and then immunohistochemistry, but we're using a multiplex immunohistochemistry called the digital space profile.
That's awesome. And what about did you ever look at the plasma responses instead of just stimulating but stimulating the PBMCs? Can you see like a systemic response ever? Because you're using a very specific type of immunization to these peptides, and with the adjuvant without?
Yeah.
Like a simpler screening method than just separating PBMCs and stimulating and everything?
No, we only did for this particular vaccine trial. We just did the full gamut of the PBMC separation, the Luminex platforms, the Luminex kits using the 10-plex cytokines, so that we had that mix of Th1 and Th2 across the 10 cytokine kit for the Luminex platform, and then based on that, you know, that was sort of enough for us to go with and just move on to really what we're really interested in, which is what's happening in the endemic populations.
I just want to correct myself. I'm not downsizing the work, but I'm just thinking because of the COVID thing, you know, you're always thinking, what is this cellular assay, the easiest one that you can do, you know, that you can measure things and try to get responses.
I think when we try to break this out or expand it in the Cambodian population, we would probably use something more high throughput, like doing ELI spots with interferon release. Because we collect PBMCs on all of our 800 children in the cohort all the time, and it's a lot of work. And so moving forward, I mean, it's good, and but doing this in Cambodia is difficult sometimes. So moving forward, we may have to, we'll probably move to different high throughput interferon assays.
Right. And the other thing that came to my mind is that with one of the first slides that you have, where you show the viral hotspots, the hotspots for virus transmission, like Brazil or like Southeast Asia, I think you're in a really excellent place to survey all of this and to be prepared for the next things to come, right? I think it'd be really interesting to see.
So it's funny, 2 years ago. Yeah. 2 years ago, or in January 2018, I was asked to give a talk to Dr. Fauci, whom everybody knows, I think, who is my boss. And in it, I was talking about what we were doing in Cambodia. And there's a Nature Communications paper from that came out in January 2018 showing where the next outbreaks would happen based on biodiversity of flora and fauna, human population densities. And, you know, the area of Cambodia is lit up. The area of China around Wuhan is lit up. And so I look back on that now and I just, you know, chuckle, since that is where we've seen the emerging viruses come. So I do think we are in a good place in Southeast Asia. And I see that Claudia mentioned, again, a biomarker in the sera of some people. So right now, that's exactly right. That's what we're doing. We're kind of having two different tracks where we're looking to have a biomarker of exposure, right, which is probably going to be more of the immunodominant proteins that we're finding. And then the other side of it is really the vaccine arm, which maybe immunodominant proteins, maybe not. And we're going to be using phage display libraries to characterize those possible epitopes, and then take it onwards for function. So that's what we would like to do, because then if we can show really, where people are being exposed, then that's easier for countries like Cambodia to target their vector control interventions and initiatives. So given it's a resource poor setting.
So I guess, so amazing, congratulations. I think everyone's had the same feeling, going through your talk. I guess we have no more questions. I just want to highlight, this initiative to have a lab outside the US, NIH based, but outside the US to study diseases in their real context of, or tropical diseases in the real context of transmission. And I think it's really amazing that you can have this laboratory set up and train people, local people, and engage local people in science in a resource poor setting, like you said. So really, really big hand of applause to that. Thank you very much, Jessica, for being with us today.
Thank you guys for the invitation.
And hope maybe we can talk about COVID in another time. So good luck with your projects.
Yeah.
And,
thanks. We'll see you in 6 months.
All
right, bye.
Take care. Thank you very much. Bye-bye.